Okay. So
Yep.
today we're going to this part, right? We're going to start this part, which is formation of embryo sac. Right? Now, in the previous class, we had discussed that when the megaspore mother cell divided, right, it formed 4 megaspores, right? Out of which only one was functional, the other 3 were not functional and they degenerated. Now, from that one single structure, we are going to see from that one megaspore, how is the whole arrangement is like. Okay, now we're going to see how the cells are arranged and put into certain places. Okay, is my voice clearly coming to you people?
Yes, ma'am.
All right. Yes. Okay, good. Now we're going to talk about the formation of embryo sac. Also, was there anything left that you people had to note down, or did you people note everything down? Was left?
No, ma'am.
Nothing was left?
No, ma'am.
Okay, good. All right. So talking about the formation of embryo sac. Now see, embryo sac is the most significant, you can say, structure with respect to the female's reproductive structure, because here only the egg would be there and here only would the fertilization take place. Now, the functional megaspore nucleus of the functional megaspore, it divides mitotically to form two nuclei. Two nuclei are formed, which move to opposite poles, forming two nucleate embryo sac. Now, depending upon the number of nuclei is the plural of nucleus, right? So, depending upon the number of nuclei, we're going to see that at which stage the embryo sac is. So, initially, when that one megaspore which was functional, it divided mitotically, because now it does not need to divide by meiosis. No reductional division is needed. Now increase in the number has to take place, and when increase in the number of cell has to take place, mitosis will come. Now that functional megaspore, it divides by mitosis and forms two nuclei. Okay, now those two nuclei moves to opposite poles. They go to opposite poles. Then later on, 2 more mitotic divisions take place to form 4 nucleated and then 8 nucleated cell stage. So 2, 4, 8 after successive mitosis. So at the end, we have the 8 nucleated stage, meaning 8 nuclei are present in one cell. That is 8 nucleated cell stage. Clear? Any doubts? Okay. Now, this type of mitotic division. Okay, no, okay, one question. What happens after karyokinesis? What happens after karyokinesis?
Cytokinesis.
Cytokinesis. What is the meaning of karyokinesis?
The division of the nucleus.
Nucleus. And what is the meaning of cytokinesis?
Division of cytoplasm.
So in normal conditions, okay, in normal conditions, whenever a nucleus divides, it has to be followed by the cell to divide. Then only 2 separate cells would be there, or else it will be a syncytium. Everyone understands what syncytium is? It's very simple. It's just the word is very fancy. Tell me, what is syncytium?
When the division starts from the cytoplasm.
No. See, syncytium is when one cell would have multiple nuclei without any cell wall formation in between them. Okay. And only one cell having multiple nuclei in it is not a normal condition. It's not normal. It's something very abnormal, you can say. Okay. Now, this type of division that is taking place within the functional megaspore, is it being accompanied by cytokinesis? Yes or no? Is it followed by cytokinesis? No, nowhere. Right now, all that I'm talking about is karyokinesis, right? Meaning the nucleus is dividing. Rest of the students, do you understand the difference between karyokinesis and cytokinesis?
Yes, ma'am.
Okay, very good. So now you're seeing that it is not followed by cytokinesis. So this type of mitotic division is known as free nuclear, meaning Three nucleus, only nucleus is dividing, the cell is not dividing. So now one cell only has 8 nuclei present in it. 8 nuclei. Okay, it's a very odd situation. Now, after this, the cell walls are formed after the 8 nucleate cell stage, meaning when the 8 nuclei have been formed, then the cell walls start forming. And this forms the female gametophyte or the embryo sac. Now, let us see how that happens. This is the whole diagram to actually explain you every single thing about it. See, initially we had megaspore mother cell, and see, everywhere ploidy has been given, whether it is haploid, whether it is diploid. Pay very close attention. Megaspore mother cell will 100% be diploid, isn't it? Because it has to undergo meiosis. Now, after meiosis has been done, then 4 haploid megaspores, each of them will be N. Getting this? Because after meiosis, 4 haploid cells are formed in general. Now, out of these 4, the 3 of them will degenerate and 1 will be the functional megaspore. Now, this megaspore will undergo mitosis, first mitosis, and form 2 cell stage. 2 nuclei will be present. Okay, not 2 cell, 2 nuclei. Why have I written cell here? 2 nuclei. Because cell is not there. There is a difference between a nuclei and a cell. So, when these 2 nuclei have been formed, they go and arrange themselves at opposite poles. In that one oval structure only, 2 poles are there, upper and lower. So it would arrange themselves according to that. Now, second meiosis will take place and form 4 nuclei. Then third meiosis will take place and form 8 nuclei. Right? Up until now, this is free nuclear division. Then what will happen? Out of those 8 nuclei, okay, out of those 8 nuclei, 2 of them will be present here and 1 will be here. So 2 synergids, 2 cells that are known as synergids. First, let us see this, then you'll understand it better. See, out of the 8 nuclei that are there, 6 of them are surrounded by cell walls and arranged into cells. The remaining 2 are polar nuclei. See now, this is how the distribution is happening. Out of the 8 nuclei that were there, okay, 3 cells are present at the micropyle end. This is the micropylar end. So 3 of the cells will come and settle here. Okay, and this set of 3 cells is known as the egg apparatus. As a whole, this set, this unit which contains 3 cells. Now see, I am saying cells. Now you see that this red portion is the nucleus, and then it is surrounded by the cell wall. Now you are seeing that this is a cell now. This is not a nuclei. But if you see here, is there any cell wall surrounding it? No. So that is a nuclei. Okay, so mind that difference. So 3 are present at the micropylar end, and this constitutes the egg apparatus. The 3 cells together make the egg apparatus. Now, what is present in the egg apparatus? So the egg apparatus has 2 synergids, the 2 cells, these like 1, 2. These are synergids. Then on top of it or here, you find the egg cell. Okay, so 2 synergids and 1 egg cell forms the egg apparatus. So a question can arise like this. What are the cells that are constituted in the egg apparatus, or what cells are present in the egg apparatus? Now, talking more about a very unique feature about synergids is that synergids, they have special cellular thickenings at the micropylar tip, which are known as filiform apparatus. Do you see this thing that looks like a lettuce? This part, the lettuce part. Yes. This lettuce is the filiform apparatus. Don't write lettuce, okay? I'm just saying this so that you people understand. Don't write lettuce. People will think the teacher is crazy or what? She has taught letters. So see, this filiform apparatus is a very unique feature of synergid. Okay, it's a very unique feature. Why? Because after the pollen tube comes, for now just understand there is a pollen tube and it will come. So filiform apparatus, it will guide the pollen tube inside the synergids. You will understand more about the pollen tube later on, but for now you understand this much, that the pollen tube will be guided with the help of this filiform apparatus which is present in the synergids. Clear? Now, tell me one thing. If this is the micropylar end, right? The tip portion, the micropylar end. Then what is the name of the opposite end of micropylar end? Very good. Chalazal end. Now, at the chalazal end, 3 of the cells will be present at the chalazal end. And those cells will be known as the antipodal cells. These 3. Do you see? Each of those are organized into cell walls, right? These 3 are the antipodal cells. So, 3 here, 3 below. The total 6 cells have been arranged. Now, 2 of them are left, right? Okay. Now, the large central cell has 2 polar nuclei. The left 2 nuclei will not be actually present as in the form of a cell. They will rather be present in the form of what? It will be present in the form of nuclei, not surrounded by any wall. Okay, so that will be called as the polar nuclei. And this whole entire region wherein the polar nuclei is present, that is known as the central cell. So polar nuclei is situated in the central cell. Is it clear? Now, they say that the mature angiospermic embryo sac is 7-celled and 8-nucleated. Now, you people count and tell me, is that justified? It being 7-celled, 8-nucleated stage. Is it justified?
Yes, ma'am.
Yes.
Okay, let us count the cells now. This is 1, 2, 3. Then this whole one is 4, 5, 6, 7. We have 7 cells. Now let's count the nuclei. So 1, 2, 3, 4, 5, 6, 7, and 8. So 8 nuclei are present and 7 cell is present. How bizarre is that, isn't it? So remember that 7-celled, 8-nucleated stage is the very unique feature of the mature embryo sac of angiosperm. This is clear? The arrangement of cells is clear? Any doubt? Very good. So a typical angiosperm embryo sac at maturity is 8 nucleated and 7 cell. Clear? I've written something here. What have I written? Okay. So this is all about the arrangement of cells, and this diagram and this diagram. This diagram though can like easily come in your question, like draw the mature embryo sac just as it is and label it. Okay, so you should know how the cells are arranged. And see, even if you draw it upside down, okay, just in case, even if you draw it upside down, but mention which side is micropyle so that the teacher would know that, okay, micropyle is, they have drawn it above, because it can be drawn like this also. Okay. Any doubts? All right, now you people can note it down and then we'll move on to pollination, which is very, very interesting. Very interesting.
Yes.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Okay.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Yes, ma'am.
Done.
Done?
Yes, ma'am.
Yes, ma'am.
Done, ma'am.
Everyone's done?
Yes, ma'am.
All right. Wait, now we'll start with pollination. Okay, is my screen visible, student?
Yes, ma'am.
Okay, okay. All right, everyone, switch on your videos. Okay. So now we're going to start with pollination. Now we saw that, okay, the male reproductive structure has been formed, the female reproductive structure has been formed. Now, fertilization is important, right? Then only a new plant species can develop. Okay. Now, for that process of fertilization, there is a process that comes that is known as pollination. Now, what is pollination? So, it is the transfer of pollen grains from where? So, from the anther after shedding, only after the anther dehisces or shed. So, after shedding to the stigma, from to where? So, to the stigma of a pistil is known as pollination. So, this transferring of the pollen grains from the anther to the stigma, that is pollination. Now, pollination is of three different types depending upon the source of the pollen or what part, like what or how many part plants are involved. So, there are three types of pollination. One is autogamy, second is geitonogamy, and third is xenogamy. Talking about autogamy, the other name for autogamy is self-pollination. So in this case, what happens is that the pollen grains are transferred from the anther to the stigma of the same flower. The flower is same, meaning the condition is bisexual. Okay. And it is observed within the same flower. Okay. Now, another thing to remember here is that there are actually two kinds of flowers depending upon whether they are exposed or not. So some plants such as the viola, the common name is common pansy, oxalis and commelina. These three plants, they have two types of flowers. One is chasmogamous flower, meaning the anthers and stigma are exposed, meaning they are open flowers. Just like if you go and see a hibiscus flower, right? So hibiscus flower would have the anther and stigma exposed. So that is that kind of a flower wherein the anther and stigma, both of them are exposed. We'll call that as the chasmogamous flower. Now, the second type of flower is clistogamous. Clistogamous means the flower is closed. You cannot see the anther and stigma clearly. So, here the flowers do not open at all. So, in such flowers, the anther and stigma lie close to each other. For the case of this clistogamous flower. Why close to each other? Because since it's a closed flower, so the flower has to be situated, the components, the male and the female components have to be situated in such a way that if the anther is dehiscing, releasing the pollen, it directly has to land the stigma. Then only fertilization takes place. So that is why in the case of clistogamous flower, the anther and stigma, they have to lie close to each other. So when the anthers dehisce, the pollen grains directly come in contact with the stigma. There are no chances of any pollination other than autogamy or self-pollination in the case of clistogamous. In the case of closed flowers, can there be any foreign pollen that comes? No, right? Because the flower is closed, there is no opening for anything else to come. So in the case of clistogamous, autogamy or self-pollination is the only way of pollination. Clear? Any doubts up till now? All right.
No.
Now, in this case, in the case of a clistogamous flower, since the pollen grains of the same flower is going to pollinate, so there is no wastage of pollen grain also because it's not going into the air, it's not going into the environment. So there is no wastage of pollens after the dehiscence of anther in the case of clistogamous or closed flowers. So they produce assured seed set even in the absence of pollinators. For clistogamous flowers, they do not require anyone to help them to pollinate because it will happen naturally. So the two kinds, and what about the chasmocamas? Here, do you think will there be a little wastage of pollen for chasmogamas?
Yes, ma'am.
Yes, there will be a little bit of wastage will be there. So autogamy is clear. Auto means self. Gamy means fusion. Autogamy, self-pollination. Clear? Any doubts in this autogamy? One thing you people have to remember is that the transfer of pollen that takes place in every type of pollination. But what is the source? The same flower is important to remember here. Now, moving forward, coming on to the next type, that is Jaitnogamy. Here, the transfer of pollen grains from the anther to the stigma of another flower of the same plant. The plant is same, but the flower is different, such as how I've drawn here. For example, this is one plant. One plant can have multiple flowers, right? It can have multiple flowers. So now when the pollen grains will be transferred from this flower to here or from this flower to here, then we'll call it as Jitnogamy. But the plant is same. This thing to remember here. The plant is same. So it is functionally cross-pollination. It looks like it is cross-pollination, but it is very similar to autogamy only. It is a form of self-pollination only because the plant is same. Now talking about xenogamy or cross-pollination. Here, the transfer of pollen grains from anther to stigma of a different plant. Now, two different plants are there and the transfer will be taking place. Now, this is cross-pollination. Clear? Pure cross-pollination. Now, tell me one thing. Out of all three different kinds of pollination, which one will create new combinations of genes?
Third one.
Third one, ma'am.
Name? Xenogamy.
Xenogamy.
Okay, very good, correct. It will bring more, you can say, unique characters. The first two, since they are autogamy only, so they will be developing just the way they are. But here, in this case, new characters may be introduced. Now, we've understood that the transfer is known as pollination. Now, transfer is actually helped or aided with the help of certain agents of pollination. And those agents of pollinations are pollinators. We call them as pollinators. Now, the pollinators can be of two types. One is abiotic pollinator, the other is biotic pollinator. The abiotic pollinators are those that are non-living, such as the wind, the water. Wind and water do not live, but biotic are the ones that are living, such as the animals. Clear this much? Okay. All right. So now the maximum number of pollinating agents that are used are biotic agents. So can you think of any agent that comes to your mind when you think about a living pollinator?
B.
Bee, honeybee. Everyone has seen honeybee sitting on flower, isn't it? So apart from taking the nectar from the flower, they are also helping in pollination. So honeybees are the best pollinators, the best. Okay. Okay, let's just keep it till here only. So people note down. If the time will be left, we'll continue. Pollination, you've understood? Any doubts in
Yes.
pollination?
No, ma'am.
Okay.
Done, ma'am.
Everyone's done?
Yes,
Yes,
ma'am.
ma'am.
Done, ma'am.
Everyone's done?
Yes,
Yes,
ma'am.
ma'am.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Oh. You can draw these diagrams also roughly so that, it can remind you.
Done, ma'am. Done.